Hello, welcome to Biology, the scientific study of life. The mammalian kidney's ability to conserve water is a key terrestrial adaptation. The osmolarity of a human blood is about 300 moles each liter, but the kidney can excrete urine up to 4 times as concentrated, about 1,200 moles each liter. Some mammalians or mammals can do even better. For instance, Australian hopping mice, which live in dry desert regions, can produce urine concentrated to 9,300 moles each liter, 10 or 9 times as concentrated as seawater and 25 times as concentrated as the animal's body fluid. Solute gradients and water conservation. In a mammalian kidney, the cooperative action and precise arrangement of the loops of Henle and the collecting ducts are largely responsible for the osmotic gradients that concentrates the urine. But even with this highly organized structure, the maintenance of osmotic differences and the production of hyperosmotic urine are possibly only because considerable energy is expended for the active transport of solutes against concentration gradients. In essence, the nephrons, especially the lobes of Henle, can be thought of as tiny energy-consuming machines whose function is to produce a region of high osmolarity in the kidney, which can then be used to extract water from the filtrate in the collecting duct. The two primary solutes in this osmolarity gradients are NaCl, which is deposited in the renal medulla by the loba of Henle, and urea, which leaks across the epithelium of the collecting duct in the inner medulla. The better understanding the physiology of the mammalian kidney as the water conserving organ, let's retrace the flow of filtrate, the excretory tubule, this time focusing on how the juxtamedullary nephrons maintain an osmolarity gradients in the kidney and use that gradients to excrete a hyperosmotic urine. Filtrate passing from Pomen's capsule to the proximal tubule has an osmolarity of about 300 moles each liter. The same as blood, as the filtrate follows through the proximal tubule in the renal cortex, a large amount of water and salt is reabsorbed. Thus, the volume of filtrate decreases substantially, but because of the salt loss, its osmolarity remains about the same. As the filtrates follows from cortex to medulla in the descending limb of the lobe of Henle, water leaves the tubule by osmosis. The osmolarity of the filtrate increases as solutes, including NaCl, become more concentrated. The highest osmolarity, about 1,200 moles each liter, occurs at the elbow of the lobe of Henle. This maximizes the diffusion of salts out of the tubule as the filtrate rounds the curve and enters the ascending limb, which, remember, is permeable to salt but not to water. Thus, the two limbs of the loba of Henle cooperate in maintaining the gradients of osmolarity in the interstitial fluid of the kidney. The descending limb produces a progressively salt filtrate, and then NaCl diffuses from the ascending limb to help maintain a high osmolarity in the interstitial fluid of the renal medulla. Notice that the lobe of Henle has several qualities of a quantar-current system, similar in principle to the quantar-current mechanisms that maximize oxygen absorption by fish gills or reduce heat loss in endotherms. In these cases, the quantar-current mechanisms involve passive movement along either an oxygen concentrations gradients or heat gradients. In contrast, The quantum current system involving the loop of Henle expends energy to actively transport NaCl from the filtrate in the upper parts of the ascending limb of the loop. Such quantum current systems, which expend energy to create concentration gradients, are called quantum current multipolar systems. The quantum current multipolar system involving the loop of Henle maintains a high salt concentration in the interior of the kidney, enabling the kidney to form concentrated urine. What prevents the capillaries of the vasa recta from dissipating the gradients by carrying away the high concentration of the NaCl in the medulla's interstitial fluid? Notice in this video, back to this one model, how the human kidney concentrates urine, the two solutes model. Notice also that the vasa recta is also a quantum current system with descending and ascending vessels carrying blood in opposite directions through the kidney's osmolarity gradients. As the descending vessel conveys blood toward the inner medulla, water is lost from the blood and NaCl diffuses into it. These fluxes are reversed as blood flows back toward the cortex in the ascending vessel. With water re-entering the blood and salt diffusing out, thus the vasa recta can supply the kidney with nutrients and other important substances carried by blood without interfering with the osmolarity gradients. That makes it possible for the kidney to excrete hyperosmotic urine. The quantum current-like characteristics of the loop of Henle and the vasa recta make it easier to maintain the steep osmotic gradients between the medulla and cortex. However, any osmotic gradients in an animal will eventually be eliminated by diffusion unless energy is expended to preserve it. In the kidney, this expenditure largely occurs in the thick segment of the ascending limb of the loop of Henle, where NaCl is actively transported out of the tubule, even with the benefits of quantum current exchange. This process, along with other renal active transport systems, consumes considerable ATP, and for its size, the kidney has one of the highest metabolic rates of any organ. By the time the filtrate reaches the distal tubule, it is actually hyperosmotic to body fluids because of active transport of NaCl out of the thick segment of the ascending limb. Now the filtrate descends again toward the medulla, this time in the collecting duct, which is permeable to water but not to salt. Therefore, osmosis extracts water from the filtrate as it passes from cortex to medulla and encounters interstitial fluid of increasing osmolarity. This concentrates salt, urea, and other solutes in the filtrate. Some urea leaks out of the lower portion of the collecting duct and contributes to the high interstitial osmolarity of the inner medulla. This urea is recycled by diffusion into the lobe of Henle, but continual leakage from the collecting duct maintains a high interstitial urea concentration. Before leaving the kidney, the urine may attain the osmolarity of the interstitial fluid in the inner medulla, which can be as high as 1,200 mosm each liter. Although isoosmotic to the inner medulla's interstitial fluid, the urine is hyperosmotic to blood and interstitial fluid elsewhere in the body. This high osmolarity allows the solutes remaining in the urine to be excreted from the body with a minimal water loss. The juxtamedullary nephron with its urine concentrating features is a key adaptation to a terrestrial life, enabling mammals to get rid of salts and nitrogenous wastes without squandering water. As we have seen, the remarkable ability of the mammalian kidney to produce a hyperosmotic urine is completely dependent on the precise arrangement of the tubules and collecting ducts in the renal cortex and medulla. In this respect, the kidney is one of the clearest examples of how the function of an organ is inseparably linked to its structure. Thanks for